Assalamualaikum and hi, we are group 1 from class CS115A. There are 5 of us in this group, which are me, Nabiha, Adlina, Ayuni, Hajar and Sarah. In this video, we will be explaining 3 articles in terms of its application of design and analysis of experiment. Displayed here are the chosen title and they are related to the field of petroleum, consumerism, and agriculture.
First, experimental study of chemical sand consolidation using epoxy and furan resin for oil wells, experimental design model. This research described the needs of experimental design in oil industry. As we all know that in oil industry, there are a lot of challenges is faced, which one of them is sand production. Excessive sand production will cause serious damage to the wellhead equipment, which will increase the cost. Therefore, this research is conducted to lower the cost of production that caused by the loose layers in oil reservoirs that due to the sand production. The increase of oil production rate, which consequently increase the drag force, is a possible phenomenon to justify the production of sand. The use of sand control method is required to produce oil from unconsolidated reservoirs. There are 3 sand control methods. The first one is oil production rate control, second mechanical method, and the third one is chemical consolidation. In this research, chemical consolidation method is used. The method was introduced from the early of 1940s, which to increase the strength of the well structure with a reinforcing chemical material which binds grains in areas of the weak structure. The purpose of this research is to present experimental investigation of 2 chemical systems of epoxy resin and furan resin in various amounts and concentration for sand consolidation. Epoxy resin and furan resin were studied to stabilize the sand in oil reservoirs. The effects of amount and concentration of the resin were evaluated on 4 main characteristics of the sample made with resin, including comprehensive strength, permeability, porosity, and Young's modulus. According to the purpose, the factor is 2 chemical system. The treatment is 2 different chemical system, which is epoxy resin and furan resin, and the response variable is the effect of amount and concentration of the resin. 2 independent variables, which are resin amount RA and resin concentration RC, with the value of 5 levels, were considered for the experimental design. The Design Expert version 7 software was used for regression and graphical analysis of the obtained data. Meanwhile, the amount of resin and volumetric concentration of the resin are determined by using the equation of RA equals to resin mass divided by sand mass multiplied with 100, and RC equals to volume of resin divided by volume of solution multiplied with 100. The conclusion for this research are by increasing the volumetric concentration and the amount of epoxy resin, comprehensive strength of the core increase, whereas its permeability and porosity decrease. Next, by increase the volumetric concentration and amount of furan resin, the comprehensive strength and Young modulus of the core increase, whereas the permeability and porosity showed parabolic behavior. In both resins, the porosity and permeability were correlated with each other.
Epoxy resin could make the core with uniform structure, and its compression strength was at 32.82 MPa. However, its core permeability or absorptive strength was much lower than furan resin. In the other hand, furan resin could produce a stronger but fragile core. Hence, it will be very difficult to work with if it is used on its own. However, the curing agents for furan resin are cheaper, easily available, and eco-friendly.
The next experimental design is experimental design research of social proof in social media shopping. The main purpose of this research is to compare the 4 social proof techniques and determine which one of them are the most effective techniques that influence consumers' purchase intention in a social commerce site. The factor is 4 social proof techniques. The treatment of the research is 4 different types of social proof techniques, which are number of followers, endorsement by celebrities, recommendation by social community, and no information of social proof. Also, treatment 4, which is no information of social proof, is a control treatment. The response variable that we want to measure is the intention to purchase on social media. Next, we continue on the importance of social proof. Why is social proof important? The first reason is because social proof is effective in increasing the product reputation. This is because the product reputation will increase if customers leave positive feedback on social media. Furthermore, social proof helps in increasing the trust of potential customer to the online sellers. The reason is because a customer will continue to buy from the same sellers if there is proof that the product is in good quality. And the last importance of social proof is social media enables seller to interact directly with buyers. For example, buyers can ask questions directly to the sellers regarding a certain product. Research procedure. Convenient sampling of university students is chosen to represent gender and the group of consumers of smartphone. Then, the respondents are randomly assigned to one of the 4 treatments. After that, the researchers gave a brief explanation about the study. Then the 4th step is the respondents are given a sufficient time to read the scenario. In the scenario, the respondents have to imagine that they need to buy a smartphone for schoolwork and leisure purposes from an Instagram seller. Their parents are giving them enough money for that purpose. The snapshot of the Instagram seller and its product was provided to the respondents. Also, the respondents were required to answer certain questions that are designed to assess their likelihood to buy from the Instagram seller. Finally, the respondents need to answer demographic questions such as age and gender. Each scenario had around 26 to 33 number of respondents. The total of respondents was 122, with 19.7% male and 80.3% female. In terms of age groups, 89% of the respondents were between the ages of 18 and 22, and 11% were in the 23 until 25 age groups. Conclusion. Results shows that high number of followers and social community recommendation plays a significant impact on buyers' intention to buy from sellers. Also, celebrity endorsement is not significantly different from not having one, which means that celebrity endorsement is not relevant in the studies. Last but not least, online sellers can choose either to increase the number of followers or to have more social community recommendation in order to increase consumers' purchasing intention.
Enhancement of plant growth acclimatization, salt stress tolerance, and verticillium wilt disease resistance using plant growth promoting rhizobacteria, PGPR associated with plum trees, Prunus domestica.
This study performed a screening of bacteria associated with the rhizosphere of Prunus domestica trees to identify bacterial strains with plant growth promoting activity. 10 strains isolated from the rhizosphere of Prunus domestica showed multiple in vitro plant growth promoting and in planta. For the experimental designs, the study used completely randomized design. Factor: 10 strains isolated from the rhizosphere. Treatment: 10 different strains isolated from the rhizosphere. Response variable is bacterial strains. What is PGPR? PGPR is a microbes associated with plant roots that promoted plant growth by provide extra nutrients, protect the plants against pathogens by affecting survival of pathogenic microorganisms, and produce plant hormone or other molecules that stimulate the plant growth and prime plants' defenses against biotic and abiotic stresses.
For the results, first one, 10 from 50 different colonies are isolated to enhance Viger index of tomato seedlings are further characterized for their in vitro and in planta PGPR activities. The second one, the bacteria treatment of tomato seeds showed that the strain enhanced significantly germination rate, stem length, and increased the number of leaves. Third, the in planta effect of selected strains were also assessed through various biochemical parameters including chlorophyll A and B content as well as nitrate reductase activity and proline concentration. Fourth, treatment of tomato seed with Pseudomonas tatsuri PR7 and Bacillus thuringiensis PR8 strains allowed for an evaluation of the ameliorative effect on plants against the salinity stress. Fifth, stem length, leaf number, leaf area, and root length as well as root fresh weight were significantly higher in bacterially treated plants under normal conditions. And the last one, Pseudomonas tatsuri and Bacillus thuringiensis showed antifungal activity against Verticillium dahliae and Fusarium oxysporum FSP melanis. For the conclusion, it can be concluded that the isolated strains of Pseudomonas tatsuri PR7 and Bacillus thuringiensis PR8 led to an improvement of growth, chlorophyll and proline contents, and nitrate reductase activity in tomato plants. Also, the plant growth promoting effects seem to be related to IAA production. They were able to alleviate adverse effects caused by salinity and the fungal pathogen Verticillium dahliae in tomato plants and help to cope with deleterious effect which occurs during acclimatization of Vitis vinifera sivim. The last one, the beneficial trait exhibited by these two PGPR inoculants toward the examined host plants under normal and stressful conditions suggest their utilization as green biotechnology which will a positive impact on agroecosystem.
Based on the studies, it was proved that the application of design and analysis of experiment has helped the respective industries to discover more chances to improve their oil production, product reputations and profitability, and also plants' resistance to diseases. That's all from us. Thank you.